Hello, everyone, and thank you for joining this professional development session. Today's presentation focuses on the ICH Q3B R2 Guideline, specifically on the management and control of impurities in new drug products. We will walk through the key concepts, thresholds, and regulatory expectations associated with degradation products. This guidance is critical for ensuring the quality, safety, and regulatory compliance of pharmaceuticals intended for global markets. This guideline was developed by the ICH, or International Council for Harmonization, and applies to new drug products that are chemically synthesized. The focus is on degradation products. These are impurities that result from chemical changes to the drug substance due to environmental or formulation factors. This guideline is not applicable to biological products, peptides, radiopharmaceuticals, or herbal drugs. Importantly, it supports submissions in the EU, Japan, and the United States. Hence, understanding this document is essential for global regulatory compliance. The primary objective of the guideline is to offer direction for the content and qualification of degradation products in new drug products. It complements the ICH Q3A R guideline, which deals with impurities in drug substances, and Q3C, which addresses residual solvents. It specifically covers degradation products resulting from interactions with excipients or packaging. However, this guideline excludes investigational products, as well as impurities from excipients or those leached from packaging, unless they qualify as degradation products. Why is this control important? Because degradation products can impact both safety and efficacy. The applicant is expected to identify degradation pathways and summarize results from manufacturing and stability studies. These impurity profiles must be thoroughly compared between development and commercial scale batches. Any degradation product found above the identification threshold, especially in stability studies, must be identified and justified unless scientific reasons prevent identification. Let's move on to analytical methods. The guideline mandates that all analytical procedures used for detecting degradation products must be properly validated, in line with ICH Q2 R1 requirements. This includes testing specificity, especially under stress conditions like light, heat, humidity, pH extremes, and oxidation. All peaks in chromatograms must be labeled and explained. Furthermore, the quantitation limit must be equal to or lower than the reporting threshold to ensure precise reporting. Now, let's discuss what data should be submitted in regulatory filings. All relevant batches, including clinical, safety, and stability, must have degradation data reported numerically. Statements like complies or meets limits are not acceptable. The results should be appropriately rounded, below 1% to 2 decimal places and above 1% to 1 decimal. Additionally, chromatograms must be included from both representative batches and validation studies. Complete documentation of batch history, manufacturing site, and analytical method used is required. When it comes to setting specifications, all degradation products likely to appear during manufacturing or shelf life must be listed. These are referred to as specified degradation products, whether identified or not. Unspecified degradation products should have a general limit based on the identification threshold. The limits should never exceed the levels at which a degradation product has been qualified. Excessive batch-to-batch variability may point to inadequate control over the manufacturing process and must be investigated. Now let's talk about qualification, the process of establishing the safety of a degradation product. If the degradation product was present during safety or clinical studies, it is considered qualified. In other cases, toxicity studies may be needed, particularly genotoxicity or repeated dose toxicity. Qualification thresholds are defined, but higher or lower thresholds can be justified based on drug class, target population, or clinical history. Reducing impurity levels through better packaging or storage may be easier than running new studies and should be considered. This slide summarizes the key thresholds. For example, if the daily dose of the drug is less than or equal to 1 gram, the reporting threshold is 0.1%. For larger doses, the threshold drops to 0.05%. Identification and qualification thresholds are further tiered based on daily intake and must follow either a percentage or absolute microgram/milligram value, whichever is lower. These thresholds are the heart of impurity evaluation and must be strictly followed. This decision tree, provided in the guideline as Attachment 3, is a practical tool. First, assess whether a degradation product exceeds the identification threshold. If it does, try to identify it or reduce its level. If that's not possible, assess toxicological risk. If necessary, conduct genotoxicity or systemic toxicity studies. The entire process is data-driven and risk-based, ensuring that unnecessary studies are avoided while patient safety is prioritized. In conclusion, the ICH Q3B R2 guideline is critical for ensuring the chemical stability and safety of drug products. It supports harmonized regulatory approaches across the US, EU, and Japan. For QA and regulatory professionals, mastery of this guideline helps build robust specifications, develop validated analytical methods, and ensure product quality over shelf life. It bridges science and compliance and is a cornerstone in the life cycle of new drug product development and registration.